Hi, this is Mr. Wartsky, and this is the second screencast of Evolution. And again, if you have questions, you can read the textbook, watch the video over again, talk to me tomorrow, and if you need to pause the video, pause the video. So this was the last slide that we were on. We're talking about Lamarck and his second law of how populations changed, of use it and lose it. So Charles Darwin, we need to always like to talk a little bit about the history. He was in med school. His dad was a doctor. His uncle was a doctor. His grandfather was a doctor. He was supposed to be a doctor, but he hated the sight of blood. Hated. And he wanted to be a pastor so he could study science. But he went to a party, which changed everything, I guess. He went to a party and he met Captain Robert Fitzroy. He was the captain of the HMS Beagle. And Fitzroy, he wanted to have a gentleman companion, basically a hired friend, because he didn't want to commit, or he didn't want to go crazy. The problem with being a captain is you were a different social class, and you couldn't associate with people that were socially lower than you. So he needed to have someone on board like Darwin, who was the same social class that he could talk to. Now, Fitzroy had reason to be concerned. His uncle committed suicide by slitting his own throat because he went a little crazy. Here's Darwin on the left and Fitzroy on the right. So Darwin said, okay, he was going to be the unofficial naturalist and an official friend. Now, Darwin's dad didn't want him to go, didn't want him to go at all. Right? So he left in 1831 for a 5-year voyage, and he found out almost as soon as they left the port that, or the bay, that he hated Fitzroy. He was a religious zealot, and Darwin was not. In fact, they found letters of apology from Fitzroy to Darwin, and they were on the same boat. Now, Darwin left the ship when he could. He made observations, collected samples, and sent them all back to England. Now, this made the official ship's naturalist, Dr. Robert McCormick, mad. He was the official doctor, and on the boat, the doctor was always the official naturalist. So he left the ship in Rio de Janeiro. He just quit and went back to England, and that left Darwin as the official ship's naturalist because there was no doctor on board. They went to the Galapagos Islands, which is a small archipelago of islands off of the coast of Ecuador, and he made observations of iguanas and plants and finches, later called Darwin's finches. He noticed that they were all similar but had different beaks due to the different environments and the food that they ate. They got back in 1831 after a 5-year voyage, and he wrote his book, and he wrote, and he wrote, and he wrote. He was a bit of a perfectionist. He was a huge perfectionist. Alfred Wallace sent Darwin and Lyell, a friend of Darwin's, an essay outlining natural selection. Now, Darwin panicked a little bit, and he published his book, The Origin of Species by Means of Natural Selection, in 1859. Since he wrote the book and not the essay, he got the credit. It took him 23 years to write this book. Now, ironically enough, Fitzroy later blamed himself for the downfall of Christianity due to Darwin's book, and then he killed himself by slitting his own throat. Now, in The Origin of Species by Means of Natural Selection, Darwin outlined his 5 principles of natural selection. So the first is that individuals of a species vary, and we know how that happens, although we will review that later. 2, some variations are inheritable. We know how that happens too. There are more individuals produced than the environment can support. Okay. There will be a competition for those resources. And 5, the individuals with the favorable traits will survive and reproduce, passing on those traits to their kids. Now, the key for Darwin was reproduction. If you didn't reproduce, then you were not fit. Those are his 5. Make sure you know them and love them because we'll be going back over and over and over again. So natural selection acted on an individual's phenotype, but genes coded for that phenotype. So individuals cannot change their traits. They can't evolve. Individuals have three choices in life. They adapt, basically they do the best with what they have. Two, they migrate, they move, or they die. And you can apply that to everything. For example, this class, you adapt, you do the best you can with what you have, you migrate, you move to a different class, or you die. Well, you don't die, you fail. I mean, dying's a little severe, I think. So Darwin does not emphasize survival. What good is it to survive if you do not pass on your genes? So Darwin emphasizes reproduction over survival. Now, survival is a part of reproduction. You have to survive to reproduce. Now, variation is the key to natural selection. It is the key to everything. Variation, and I will say it all the time, variation in the population. So, what are the sources of variation? Well, we have mutation. Yeah, okay, that's a very small source. We have sex by recombination of the chromosomes when they cross over, okay? The independent assortment or the shuffling of the chromosomes and the joining of two unique gametes will give us a unique individual. And then there's migration. Migration introduces new genes to the population. So migration is the populations that have migration are probably the strongest evolutionarily. So how is variation maintained? Well, sex, got that, recombination, independent assortment, two unique gametes, right? Outbreeding, which is the opposite of inbreeding. You breed out, right? You pick individuals that have different phenotypes. Diploidy, you have two genes per trait, at least two genes per trait, which can lead to heterozygote superiority, which the heterozygote has an advantage over the homozygous dominant. So this maintains the recessive gene. So if we take a look at sickle cell, those individuals that are heterozygote and have the sickle cell allele are resistant to malaria. Thalassemia, if you have the recessive thalassemia allele, you are resistant to malaria. If you have the allele for Tay-Sachs in the recessive form, you have a resistance to tuberculosis. Cystic fibrosis, if you're heterozygote, you will be resistant to typhoid. It is thought that every recessive anomaly, there's heterozygote superiority. That's what is thought. So how do gene frequencies change? Okay, all the genes of a population, that is called the gene pool. And if in evolution, you will see a change in the gene pool of a population over time. Now, these changes can be either random or non-random. Now, let's talk about the random changes to the gene pool. Everybody in? So the first random change to the gene pool deals with gene flow, and that's basically immigration or emigration. Every time we have an influx of immigrants into the population, our population gene pool changes, and that's good. Same thing if individuals leave, our population gene pool will change. Genetic drift are changes to the gene pool just by chance. So the founder effect is when a small group leaves, breaks off from a larger group and leaves and goes and populates somewhere else. Now there's a change in gene frequency for the smaller group, and there's going to be a change in gene frequency for the larger group. So both populations will evolve. We have examples of that with the Pilgrims, with the Quakers, the Afrikaners. These individuals leave and go somewhere else. Population bottlenecks is when a disaster happens and leaves a small population behind. So a large number of the individuals die, but it's a chance if you survive. Non-random mating. So this is mating based on phenotype. So this causes the gene pool to change basically by increasing homozygosity, which is bad. This is known as inbreeding, and we have populations of people that will not breed out of their ethnicity or religious order, and that increases inbreeding and increases homozygosity. So you need to mate out of, in order to increase variation, out of your phenotype. And then we have mutations, which is a really small way to maintain or change the gene frequencies of a population. So non-random changes to the gene pool is basically natural selection. Organisms with the better traits to obtain the limited resources, they survive and pass those genes on to their offspring. The other organisms that could not survive, they either moved to a different area or they died, but their genes were removed from the gene pool. How can we measure the changes to the gene pool so that I can show you that evolution happens? Well, we are going to calculate the gene frequencies in a population. And if we see changes of the gene frequency in the population, we will have witnessed evolution. And the equation that we're going to use is the Hardy-Weinberg equilibrium equation. So pay attention to this. So we're going to use big A and little a for the alleles and the letter F for frequency. In a population, there are three genotypes. There's big A, little a, and little a, little a. If you're in a population, you have to be one of those three genotypes. So the frequency of big A plus half the frequency of big A, little a is equal to the frequency of big A, right? Frequency of little a is equal to the frequency of little a plus half the frequency of little a, little a. So if we say that P is equal to the frequency of A and Q is equal to the frequency of little a, P will therefore be equal to the frequency of big A plus 1/2 the frequency of big A, little a, and Q would be equal to the frequency of little a plus 1/2 the frequency of, that should say big A, little a. Write down big A, little a. That was my typo. So therefore, we can say that the P plus Q is equal to the frequency of big A plus the frequency of big A, little a plus the frequency of little a, and that is equal to everyone, so that is equal to 1. Therefore, we can say P plus Q equals 1. We can also say that 1 minus Q equals P, or 1 minus P equals Q, right? So the probability, okay, so hopefully we got to understand P plus Q is equal to 1. So the probability of A meeting an A is equal to the frequency of A times the frequency of A, which is P times P, which is P squared. The probability of meeting a little a is the frequency of little a times the frequency of little a equals Q times Q, which is Q squared. The probability of a big A meeting a little a is equal to the frequency of big A times the frequency of little a, which is P times Q, which is PQ. And it is the frequency of a little a meeting a big A, which is Q times P, which is also PQ, so it's 2PQ. So then we can say that in a population, P squared plus 2PQ plus Q squared is equal to 1. Let that sink in. So now we have two equations, P plus Q equals 1, and P squared plus 2PQ plus Q squared is equal to 1. So if there are two phenotypes, dominant and recessive, if you are dominant, you can either be big A or big A, little a. That makes sense, right? If you are recessive, you have to be little a, little a. We know your genotype if you are recessive. So we can calculate the frequency of little a, or Q squared by counting the number of people with that trait and dividing by the total number of people. If I lined up 100 people and had everybody stick out their tongue and try to roll it, if 25 people could not roll their tongue, I know that 25 out of 100, which is 0.25, that is my frequency of little a, or Q squared. So Q squared, therefore, would be 0.25. If I know Q squared, I can take the square root of Q squared to find Q. So the square root of 0.25 is 0.5. If I know Q, I can calculate P. How? Because 1 minus Q is equal to P. So P would therefore be 1 minus 0.5, which is 0.5. Now, if I have P and Q, I can calculate P squared as well as 2PQ. P squared would be 0.25 because that's 0.5 squared, and 2PQ would be 2 times 0.5 times 0.5, which is equal to, guess what, 0.5. So in my population of tongue rollers and non-tongue rollers, 25% would be big A, 50% would be big A, little a, and 25 would be little a, little a. I hope you got that. Have a good night.